Hello everyone and welcome back to Biology with Mrs. Evans. In the last video we talked about simple Mendelian genetics, traits that were controlled basically by two types or two versions of that specific gene, and dominant versus recessive, etc. Again, remember Gregor Mendel studied pea plants, and he used pea plants to learn a lot about how traits were being passed from one generation to the next. And again, he did his work in the 1800s. There were lots of scientists that came after him that used his ideas to come up with different experiments. And we know that there are differences between plants and animals. So a scientist questioned whether or not the modes of inheritance were the same for plants as they were for animals. So Thomas Hunt Morgan is a name that you also need to know, and I'll write it here on the board. He was a scientist that studied fruit flies. Fruit flies, for the same reason that Mendel chose peas, he could easily manipulate them to control what type of fly crossed with another. He could also generate lots of offspring in a short period of time, and he could also look at several different generations again in a short period of time. And Thomas Hunt Morgan confirmed basically a lot of what Mendel did, but he and other scientists came up with some problems because not all of the traits that they studied showed basically the genetic patterns that Mendel had described. And the four areas that we're going to talk about is what happens when there is incomplete dominance, codominance, polygenic traits, or sex-linked traits. And we're going to talk about each of those in a moment. So remember, you need to know Thomas Hunt Morgan studied fruit flies, and he confirmed pretty much most of what Mendel did. Again, he came up with these exceptions for traits, and his work, and as well as other scientists, we basically expanded upon modes of inheritance, or how traits are passed down from one generation to the next. So incomplete dominance. Incomplete dominance simply means basically that a trait is not exhibited in a normal fashion. For example, a red flower crossed with a white flower, all of the offspring would be pink. And what that means is, for incomplete dominance, is that there is a masking or a blending of the two. So if you take a red marker and a white marker and mix the two, or red paint and white paint, if you've ever taken an art class, you know that that color comes out pink. So in incomplete dominance, an individual that is heterozygous will show a blending of the two alleles instead of one being completely dominant over the other. So that is incomplete dominance. A heterozygous individual will show a blending of the two traits. So typically we write it as you see here on the board. All right, red is big R, and then white would be big W, and then an individual that is heterozygous will show the pink, or the blending of the two. So RW will show pink flowers. The same would be true. Let's say we have butterflies, and they are black is dominant, and we cross a white butterfly as well, and all of the offspring come out gray. All right, that would indicate that that is an example of incomplete dominance, because notice that all of the offspring are heterozygous and all of them are gray, a blending of the two. So that would be an example of a trait that would exhibit an incomplete dominance inheritance pattern. Then you have crosses where you cross a red flower and a white flower. So go back to what we had a second ago. A red flower and a white flower. Now the genotype is the same. And usually when we write these, we kind of put them in alphabetical order. So R comes before the W, so that's how we would write it. So when you cross a red flower with a white flower, all of the offspring come out looking like this. And this is what we refer to as codominance. Both traits are equally dominant to each other. Not one of them is not more dominant than the other. So when codominance is present, you're going to see both traits in a heterozygous individual. So notice all of the flowers from this cross, if it was a example of codominance, would show this red and white pattern that you see here for this flower. So sickle cell anemia. You heard me talk about this earlier in the year when we looked at red blood cells underneath of a microscope. Remember, they had this kind of disc shape and it was kind of concave here. That is a typical red blood cell. But when you look at individuals that have sickle cell, the red blood cells, remember, because of a mutation with the hemoglobin, ends up looking more like a sickle or a scythe that you see here. So an individual that is heterozygous for this trait, they're going to have both normal and sickle cell in their blood smear that you would see a doctor basically look at. So here's an example of an individual that is heterozygous for that trait. Notice that they have normal red blood cells that you see here, and then you have the ones that are misshapen here. All right, so this individual is heterozygous, and this is an example of codominance, where you're going to see both traits expressed at the same time. Polygenic traits. Polygenic traits are traits that are controlled by more than one gene. These traits usually show a wide range of differences in individuals, and one of the ones that you guys always ask about is eye color. Right now, as it stands, we know that the human eye color is controlled by at least 15 different genes. So you can see all of the variations that you see on this slide is basically a combination of those 15 genes. And eye color is one, skin color is another. You'll notice all the different hues to individual skin. Your hair color is another, because they're not all red or blonde. There's some differences there. So polygenic traits are traits that are controlled by more than one gene. And then what we call sex-linked traits. So we talked about autosomes. Remember, the first 22 pairs of chromosomes for humans are what we call autosomes, and then the 23rd pair is what we refer to as our sex chromosomes. And I already talked about this earlier in the year. Females are XX. That means that we are homozygous for this, and then males are XY. So if you look at basically the chromosomes that determine sex, this is what it would look like as you see here on the screen. This would be a female's, and then this would be a male's. So what we know is that there are certain traits that are controlled by genes that are located on the sex chromosomes. Most of them are going to be controlled by the X chromosome. There are a few genes that are controlled by the Y chromosome. So for sex-linked traits, we look at what chromosome they're being basically the gene is on and how that's going to be passed down, and we're going to talk about how to set those up in a moment. But what's an example of something that is sex-linked? Colorblindness is an example of a trait that we find on the X chromosome. It is a recessive trait. So individuals that are colorblind either have two versions of that gene if they are a female, and then for males they only need one version of that gene to be colorblind because they only have one X chromosome. So one of the things that you need to know is what are the chances for a couple to have a boy or a girl baby. And you need to know that it is the father basically that determines the sex of a child, because remember, females are XX. As you see here on the screen, the mom can either give a big X or a big X. It's the dad that determines the sex because they can either give an X chromosome or they can give a Y chromosome. If they give an X chromosome, the child is going to be a female, and if they give a Y chromosome, the child is going to be a male. So the probability is always going to be a 50-50 chance. All right. Sometimes you'll see a question of a couple that has had 10 children and all of them have been male, and they want to know what's the probability that their 11th child will be male. It's still 50-50. It doesn't increase to 100% because all they've had is males. It's always 50-50 because the chance is either giving up an X or a Y, and it's a 1 in 2 chance, so it's always 50-50. So don't be tripped up by a question that is similar to that. So sex-linked traits, again, traits that are located on the sex chromosome. The majority of them are going to be on the X chromosome. There are very few that are on the Y, but we're going to talk about modes of inheritance for that in a second. So colorblindness, it is on the X chromosome, and it is a recessive trait. And if you go into the military, you'll have to do a physical called MEPS, which is Military Entrance Processing System, I think it is. I had to do it when I was 19, when I thought I was going to join the military. And you'll get a series of cards that look like here, that you see here pictured on the screen. If you are colorblind, then you will not be able to see the numbers that are in this particular screen. So you'll see there is a 4 here, and then there is an 8. If you can't see those numbers, then that indicates that you are colorblind for a specific color. You'll notice that the 4 is green, and then the 8 is blue. So my husband, he is colorblind. If he were to look at this slide, he wouldn't be able to see the numbers that are there. So sex-linked traits, we're going to look at how those traits are passed down here in a second. So hemophilia is another X-linked disorder that is recessive. So again, for females, in order to show a X-linked trait, we would have to be that is recessive. We would have to have both copies of that allele. And then for males, because they only have one X chromosome, it would be they would show the recessive trait if they had that allele. So for sex-linked traits, how do we determine crosses for that? So let's look at colorblindness. So my husband, he is colorblind. So I know his genotype. Because he is a male, his genotype is X, Y. And the way that we denote the mode of inheritance is if it is recessive, again, you're going to use a lowercase letter. So I'm going to say that normal vision is an R and colorblindness is a little r. So my husband's genotype for that trait would be X, little r, Y. I am not colorblind. All right. Now I don't know for 100% certainty what my genotype is. Okay. So I know I'm not colorblind, so I know my genotype is at least big R, little r. All right, so we're going to see if I was heterozygous, what are the chances that I would have a child with my husband that is colorblind? So the same rules apply. You're going to distribute the two. So you're going to have X, capital R, X, R, X, R, X, R. So if I was heterozygous, then as you can see from the results, there is a 50% chance that our child would be colorblind. 25% colorblind female, 25% colorblind male. 50% that our child would not be colorblind. Now, I don't know for sure what my genotype is. So let's say that I am dominant, or I'm homozygous dominant. So again, we'll do the same cross. My husband's colorblind, so he is this. So the chances of us having a colorblind child if I was dominant would be 0%. So some terminology that you need to know for this. We refer to individuals like this child of ours, if I was heterozygous, this individual would be what we refer to as a carrier, meaning they don't show the trait, but they have the gene that can be passed down for that trait. So if I was heterozygous, I would be a carrier because I could pass that gene on to my child. So this daughter of ours would be a carrier for colorblindness as well. So sex-linked traits, you need to know how to practice those. We're going to do a lot of those problems together in class. Let me bring up another slide here in a second.